Okay, so welcome to today's seminar. We're very lucky to have our speaker today, Peter Walter, visiting us. I'm sure to many of us sitting here, Peter needs very, very little introduction, but regardless, I'll do a very quick introduction of Peter. So Peter graduated from the Free University of Berlin in 1976 and received his Master of Science in Organic Chemistry from Vanderbilt University in 1977. Only 4 years after that, he obtained his PhD in biochemistry at Rockefeller University, and only 2 years after that, he became a faculty member at UCSF Department of Biochemistry and Biophysics, and he stayed there for almost 40 years. So recently, Peter retired from UCSF and joined the Altos Labs, Bay Area, and he was the founding institute director of Altos Labs from 2021 to 2023. So over the almost four decades of scientific research, Peter's labs at UCSF and Altos Labs made numerous discoveries in proteostasis networks, including the unfolded protein response, the integrated stress response, and so on. And his work has been recognized by many awards. Just to name a few, there's a Shaw Prize relevant to us in Hong Kong, and that happened in 2014. The Lasker Award, the Wilczek Award, and the Breakthrough Prize. So actually this week, Peter's in town as Shaw Laureate for these Shaw Forum activities. So that starts tomorrow. If some of you have registered for these events, you have more opportunities to interact with Peter. So before Peter begins, I like to take this opportunity to have my 2 minutes of spotlight over here. So having been a postdoc in Peter's lab for 6 years, I have always envied one thing that happened to students in Peter's lab. So at UCSF, students did not have a thesis defense. Instead, they had an exit talk. So this is more like a relaxed format of talk where you kind of summarize your work and is attended by parents, friends, and everyone. So before that, Peter will always give a very quick summary of the students' time in the lab over a number of interesting pictures. And as postdocs, we did not have this opportunity. We did not have an exit talk. So I always envy that. But now it's my opportunity to do this for Peter. So I only have 2 pictures to show, okay? So I mean, I had really nice time in San Francisco, and a lot of my memory there is tied to my time in the Walter Lab. And I think over the many, many lessons I learned from Peter, there are 2 that really stuck with me. And the first thing is to always keep the fun in science. And Peter's always a very fun person, always almost the only person who dresses up in Halloween in the entire genetic hall. Okay, so for the lack of better pictures, okay, all right, that one. So this may not make too much of sense to you guys. It looks like just a regular bench. So this the interesting thing about this is there's a red carpet, and the bench the red carpet is leading to was actually my bench at UCSF. So this picture was taken, I think 2020 in the middle of COVID. So COVID doesn't look very relevant these days, but back then it felt like the end of the world to many people for different reasons. For me personally, I was stuck at home in Beijing for 8 months during COVID because my visa couldn't get through due to all the travel restrictions. So as I stayed in Beijing, the longer I stayed, the less hope I had. I almost, in the end, gave up and thought I would never be able to return to my postdoc job. And this was in the middle of my postdoc, so that was really bad. But so one thing I think one lesson I learned from Peter is always to be optimistic and stick it out to the end. So Peter not only helped me accelerate my visa process and basically rescued me out of my parents' apartment in Beijing and return to the lab at UCSF, and I also get this red carpet celebration. So that was me in my mask. Back then we had only 25% occupancy. Everybody can't go to the lab all at once. So I took this alone. But anyways, that was something I still remember today. So Peter is not only one of the best scientists, but a great, great mentor. So anyways, that was my 2 minute. And Without further ado, let's welcome Peter.
Well, thank you, Lan, for this warm introduction and the personal touch. And thank you all for rolling out the red carpet here for my visit in this wonderful place. So you all know Lan, and as you can imagine, the schedule she made for me this morning was quite dense, and I had sort of 5 minutes with 200 people to exhaust me entirely. But I really enjoyed the science, and you have a wonderful place. So and I hope you all realize that and have the fun in science. So I hope I can reflect that in the science I'm going to talk to you about today. It's been a really fun project. I'm going to lead you all the way from discovery to something very personal at the end. And it's been an amazing ride. And what I would like to tell you about is not just the science, but also a little bit of the path of discovery, how we got there. There are many messages in there for particularly the young students of how that this is not just a job. It's an adventure. And I would like to start out by telling you about a dream. And I think we all should have dreams of where we are going with our science. So just be with me for a second. What if we could figure out what defines a young, healthy cell and devise a way of maintaining the cell health longer in life? We could use this knowledge then to approach biomedical research in disease-agnostic ways, right? Bringing everything back to a young, crispy state with which we were originally born. And in a way, if we understood this, we could take our current system, which is really not healthcare, but it's sick care. We go to the doctor when we have an ailment and nudge it towards an actual healthcare system where we maintain our health in the 10 to the 13 cells that we are each made up of. And with this in mind, we've been studying now for many, many years the cell stress pathways, which have this remarkable feature of regulating cell health by controlling homeostasis. And it's the cell stress pathways that sense deviations from homeostasis. And for discussion's sake, I would like to define a young state as a homeostatic state where everything is in balance and works perfectly. Just as if you have a little baby, right, and it scratches itself, then the next day the wound is gone. It has healed. There's no scar. Everything is just repairing optimally and functioning optimally. Stress pathways, by sensing homeostasis and the deviations from it, they selectively mark sick cells. Right? They're activated in cells in which the homeostasis, the balance of components, not just proteins, but metabolites, lipids, RNA, is not working perfectly. Therefore, they don't really matter as much in healthy cells. So when we inhibit them, when we manipulate them in the lab, we find that that these manipulations are often not very toxic compared to other means of like chemotherapies. They are very important. They make life-death decisions. So the reason why not all of us have cancer is that these stress pathways are activated in cancer cells. A cancer cell develops right in the niche of a body. It evolves to become ever more aggressive and not following the rules of the body anymore. But this evolution is not directed. So they become genomically unstable, and all the changes that happen in the cell are not necessarily helping it to become more aggressive. So they make many mistakes, many mutations, passenger mutations, make protein complexes in the wrong proportions. Right, and all of this activates the stress pathways, which in this case get used by the cell to band-aid over these defects that normally would lead to apoptosis. So in cancer, this plays a major role, that this decision between life death is a major thing which we can influence and potentially do very good in that disease category. And of course, it's also from a research aspect, it's a fascinating question, right? How does a cell make a decision? Can I fix something or should I kill myself? If you come up with molecular answers, they are very exciting by definition. And then we find that these stress pathways are often dysregulated in disease, or diminish or shut down with age. And as such, they affect many numerous age-related diseases. So we've been approaching this pathway as an incredible resource for doing disease-agnostic studies and seeing where we can open therapeutic windows to bring things back into homeostasis or make the right decision. And this affects entire disease classes, so not just a particular cancer, but pretty much many. It's defined in many in entire classes such as cancer, cognitive disorders, neurodegeneration, diabetes, metabolic disorders, inflammation, and we could make that list. We could extend that list into many other diseases that we find out there, particularly age-related ones. So what can we do about it? So I'm going to give you one example just from the cancer arena, and that's a long-standing collaboration that we have with Avi Ashkenazi's lab at Genentech. And in this experiment, here we are taking a mouse, we are injecting in it a human cancer cell line, a very aggressive blood cancer. You establish this tumor in the mouse, and then 2 weeks later, that tumor has spread all over the animal, including the metastasized to the brain. And it's basically it's still a pretty much uncurable cancer. Now what we did is we do the same experiment, but in this case we use an inhibitor against one of these stress pathways, the kinase IA1, which I'm not going to talk much more about, but it's one of these key sensors of misfolded states in the endoplasmic reticulum, which is enhanced in cancer cells. So the unfolded protein response, of which IA1 is one of the sensors, is upregulated in these mice, in these cancer cells. So we first establish the tumor, then we inject an IA1 inhibitor in that, and this is what we get 2 weeks later. So basically, not only does the tumor not grow or metastasize anywhere, it is the existing tumor dies and is eliminated. This is not just true for multiple myeloma, as I told you. Similar experiments are obtained in triple-negative breast cancer, not just by us, but Lori Glimshaw's lab. So it's extendable into other cancers. And this is now something that's in clinical development, preclinical development, as a new way of chemotherapy. It's not toxic, hardly any side effects. So compared to conventional treatments and therapies, it's synthetic, or it's synergistic with existing standard of care. So you can use now much lower doses of bortezomib or proteasome inhibitors, which are much more toxic, and add this inhibitor on top of it, and you get very beneficial effect. So we hope that this will open a totally new arena for chemotherapy approaches that would be broadly applicable, and not just be specified to one particular cancer, but be broadly applicable to this whole category of different diseases. So I will spend the rest of my time here talking about cognitive disorders that we, as you'll see, we got into without ever ever wanting to go in that direction. But this is simply where the meandering path of discovery led us. And the primary observation that this is all based on, the main discovery, was made some 15 years ago, 20 years ago, by Mauro Costamaggioli when he was a postdoctoral fellow in Nahum Sonnenberg's lab at McGill. And what Mauro discovered is that this step of long-term memory formation requires ongoing translational activity in the cell. This was long known from studies from Cadell and others learning in aplesia. But what Mauro found is that this translational step here is regulated by a particular translational control event in the cell that is the center of which is the phosphorylation of this very generic translation initiation factor ERF2. I'm going to tell you much more about that thing as I go along. Suffice it to say, we have phosphorylation of a single serine residue here, which blocks translation, and by blocking translation, then does no longer allow long-term memories from forming. They made this discovery based on genetic experiments, in which they took mice and they mutated this single phosphorylation site to an alanine, the serine. And in the heterozygous mice, they made the observation that these mice suddenly learn better. They're smarter. The homozygous mouse is dead. But only being able to make half as much of this phosphorylated form improves the ability to store long-term memories. So based on that observation, which, by the way, this regulation event here is called the integrated stress response. Again, I'm going to come back to that. We happen to have in the lab an inhibitor of this regulatory event, which is called ISRIP, so a small molecule that blocks the effects of this phosphorylation. And because it now blocks the effect of the phosphorylation, translation cannot be inhibited. We were simply asking the question, well, are these mice learning better if we inject this compound into them? So we send it to Nahum's lab, and they put it through the paces of this learning scheme, where you have this classical Morris water maze. Mice in this trough of milky water have to remember where to find this hidden platform here. They orient themselves by literally posters on the wall in this trough, and they learn how to find this platform. And they then, in this training period here over a couple of days, you see that they plateau at a certain performance of the time it takes them to find the platform. Now, if these mice were injected with this ISRIP, their learning now is substantially improved. So this is, I think this is already a very, very remarkable finding, because it basically tells us that the integrated stress response via this phosphorylation event inherently limits memory formation, and that's with this drug that inhibits the integrated stress response with ISRIP, we can release this brake. It's remarkable because we're improving here on a wild-type animal, on behavior, right? So evolution hasn't optimized this process, and we can come back in the discussion why this might be the case. But it basically says that ISRIP is a cognitive enhancer under these conditions, which of course has lots of room for abuse. And there are these websites that you can find where people are engaging in self-experimentation and brain doping. So really Darwinism in action here in our societies. You can buy it from Alibaba. They quickly realize that this is something they can market, but that's not where we want to go. So we were wondering, right? Can this open new paths to explore potential therapies under conditions with cognitive defects? So we teamed up with Susanna Rossi, who at the time was running the trauma center, or running a lab in the trauma center at UCSF. And she has this modified version of the Morse water maze here, testing animals with traumatic brain injury. So mechanical injury to the brain basically leads to dementia and the inability for these mice and for humans to learn. So here's a mouse, the control mouse here. It has learned how to find this platform. You see it swims a direct path, whereas this mouse here, subjected to this mechanical injury, is basically unable to learn. It swims a random path, and it takes forever to find the platform. It eventually finds it, so the animal is not tortured in these experiments unnecessarily. So here's the experimental protocol for the data I'm going to show you. We inflict the injury in the mice, a mechanical injury to the brain. We then let the mice recover for 4 weeks. And after then, every inflammatory condition is pretty much subsided. The mice swim at the same pace. So they're perfectly fine, but they cannot learn. They're fully demented. Then 4 weeks after, we inject the drug. We subject these animals to a training period. After 3 of these injections, we do a memory test. We let them sit for a week. We do another memory test. And if we would not intervene with drug treatment here, they would remain demented and not able to learn for months to come. So here are the data. This is day 1 of the 4 weeks after the injury, the first day of training. You see that the injured animals do not learn. They make the same number of errors, swimming into the wrong arm. Whereas the control mice get better over the training period. They make fewer and fewer mistakes. The next day, the injured animals remain demented. The control animals remember what they've learned the day before and hardly make any mistakes anymore. A day later, they remember, and they keep on remembering. Whereas these guys stay flat. So there was no drug here. There's just the controls to set stage for these experiments. So when we inject the drug now in the regime I told you about, already on day 1, you see this marked improvement of learning as the training progresses. And on day 2, it's a miracle. The injured mice now become indistinguishable from the control group. So they've basically learned as well as the control group. A day later, they remember. A week later, they still remember. So this is highly statistically significant, and it's I find it incredibly remarkable. This is one way of cognitive defect. We can look at different models. One is a chromosomal imbalance that is inflicted in individuals having Down syndrome, where you have extra copies of a chromosome. This gives you induction of stress pathways because now proteins are assembled in the wrong proportions in the complexes. They are being seen as abnormal. Stress pathways are activated. You get cognitive defects in this mouse model, and again, it is cured by injection of this drug. And genetic manipulations that are of the kind the heterozygous have phosphorylation event. So everything pretty much I tell you about the drug is paralleled by genetic experimentation that shows the same result. The curves are virtually superimposable. We can take old mice which have difficulties learning, we inject the drug, and we can turn them back into teenagers. Very remarkable. Let me show you, actually, aging may not always be such a bad thing, right? So this is an example here of me as a graduate student before Lan ever made a decision of joining this lab. Let me show you one more essay here. and that is it's a mouse model for something like risk taking or anxiety. And what you do is you just put the mouse in the middle of the structure here, which is basically 4 arms. Two of them are shielded, so the mice is a nice protected space, or it can decide to venture out onto these arms, be exposed to the light, and doesn't could potentially fall down, but then we have some cushions down there so they don't hurt themselves. And the experiment here is to subject the animals to repetitive mild brain injuries. These are basically concussions. The effect of a concussion, like football players would experience, are additive. So repeating it makes it worse every time it happens. And again, the mice after these five concussion-like treatments here are entirely demented. We again let them recover for 4 weeks. We inject the drug, put them into this elevated cross maze here, and then we let them sit for another 3 months. No more drug injection. And then we do the same assay. The drug has a half-time in the animal of 8 hours. So under these conditions, we cannot detect it anymore, and it's not plausible that there's drug left in the system. Here are the data. So animals that have been subjected to the concussion treatment here spend much more time in the open, so they're much more risk-taking or have less anxiety. Injection of the drug entirely reverses this phenotype. If we now look 4 months later after the injury, you see that the effects of the injury have lasted, but the effect of the drug that was injected now 3 months earlier have also lasted. Right, so it is as if we had had hit the reset button, and effectively cured the effects of the treatment. So this is probably the most important slide I'm going to show you. It basically summarizes all these different conditions that have been used to assess the system. In all of them, under all of these conditions, we know that the integrated stress response is activated as manifested by phosphorylation of the translation factor. In all of these conditions, we show that the brain cognitive function is impaired. So we have all the neurodegenerative diseases, Alzheimer's, frontal dementia. I've shown you about Parkinson's disease. I've shown you traumatic brain injury effect and Down syndrome. We have age-related cognitive decline. And also it has some inherited diseases like vanishing white matter disease. I'm going to come back to that and show you a little bit later. Under all of these conditions, is the integrated stress response induced? And if we prevent it from taking action by either the genetic manipulations or adding this inhibitor here, we inhibit the integrated stress response and we restore brain health. So in other words, we really think that it's the integrated stress response, not the particulars of these diseases, which all have very different molecular causes, that are the culprit for the broad spectrum of these cognitive defects. I've also shown you that for the traumatic brain injury here with the long-lasting effects of the drug, that there must be some maladaptive feedback loop here, that once the response is on, it keeps itself on, and therefore we get these long-lasting effects of the injury. And if we block that feedback loop by injecting the ISRIP, we'll also restore, and we basically reset the system under conditions where the insult is just happening at one point in the time at the time here. And this is not just cognition, right? Because the assay I showed you with this elevated cross maze is basically, there's no learning involved. You just put the animal in the middle and you see what it wants to do. So that's innate behavior that was affected by the brain injury and that has been restored to normal conditions by inhibiting the integrated stress response. So this is all good news for the mice in the audience. Of course, human brains are much different from mouse brains, but nevertheless, we are very hopeful that some aspect of this will translate. The industry has taken note of it. The ISRIP analogs are currently in clinical development. So ISRIP analogs passed phase 1 AB trials in both analogs made by Calico AbbVie, to whom we licensed, UCSF licensed the ISRIP molecule, and then in competition, other companies have picked it up. Denali is one of them, and it's currently in human trials in individuals afflicted with vanishing white matter disease and major depressive disorders, simply for reasons that the clinical hurdle of these conditions is the easiest to jump over and get the stuff into the clinic. So ISRAP in humans is, or the analogs here, which are medicinally improved, is safe in these patients. So all of this basically serves to get you interested in the rest of what I'm going to tell you about, and that is, what is ISRAP and how does it work? And what can we do with that knowledge that we have accumulated over the years? So I told you already, the ISRIP, the integrated stress response, involves that translation factor, EIF2. EIF2 is a trimeric protein that is a GTPase. And like most GTPases, it switches between a GDP bound state and a GTP bound state. And when it's loaded with a triphosphate bound state, it picks up methionine initiator tRNA, and that brings the methionine to the initiating AUG in the mRNAs that are being in the transcriptome. So it's a very generic translation initiation factor, pretty much involved in every mRNA translation that starts with an AUG. Like from most GTPases, this exchange is not spontaneous, but rather is carried out by an enzyme, nucleotide exchange factor, that is, in this case, a huge complex of 10 subunits, 5 different kinds, 2 each, so it's 2-fold symmetric. It catalyzes that exchange reaction to then make translation happen. Now, when ERF2 becomes phosphorylated at the single serine residue at the alpha subunit, it now sticks more tightly to the exchange factor and pulls it into a state where this reaction can no longer be carried out. So it becomes an inhibitor. So we convert the substrate to an inhibitor. And since there's 10 times more of the EIF2 than there is of the exchange factor, a little bit of phosphorylation can go a long way of pulling this exchange factor out of this reaction. Now, this phosphorylation event is carried out by at least 4 different kinases in the cell, and each of these kinases senses different stress conditions in the cell. So we have PERK, a transmembrane protein of the ER, part of the unfolded protein response, that responds to unfolded protein status in the ER. We have accumulation of double-stranded RNA in the cytoplasm. Viral infection activates PKR. Nutrient conditions activating GCN2. So amino acid starvation or redox conditions or mitochondrial health is being read out by heme deficiencies, read out by HRI. And all of these kinases converge on the same serine residue for phosphorylation, and that's why it was named the integrated stress response, because integrating between all these different stresses. Now, the consequences then of this phosphorylation event is that a general inhibition of translation, But seemingly paradoxically, there are also some mRNAs that are now selectively translated. And these encode some proteins that are present in pretty much every one of our cells, ATF4, a transcription factor, or some proteins that are tissue-specifically expressed, like OPHN1, which is a neuronal transcript. And the reason why these mRNAs escape this translational block is that they contain this particular structure in their 5' untranslated region, where small open reading frames are being translated. Their translation prevents the actual AUG from being reached. So if translation cranks and initiation happens efficiently here, no ATF4 is being produced. But if we now have a phosphorylated state and therefore translation initiation is lowered, these open reading frames are used differently, so that now access to the AUG down here is allowed and ATF4 is being produced. So we took this particular biological phenomenon, which was, by the way, worked out in yeast by Alan Hindenbusch. So there's a long history here, and yeast as a model system is really, really powerful. And we changed the ATF4 reading frame here for a reporter to now screen for small molecules that would inhibit this response. And this was done in the lab by a former postdoctoral fellow, Kamela Sidrowski. Kamela, by the way, then went on to become a scientist at Calico and developed and now leads the clinical development of the drug. And she screens her library of some 100,000 compounds in an academic setting that for us seems to be a lot at the time. Sounds very easy, right? You put luciferase reporter in the cell. When the integrated stress response goes on, the light goes on. You look for molecules that turn the light back off. Took us 3 years to sort through 100,000 compounds. And in the end, Kamela ended up with one. And this is our ISRIP molecule that I told you about before. Now, it turns out that the screening facility with which we collaborated at UCSF triaged against this compound and said this would never make a drug, don't pursue it any further, we throw it away. So everything I told you about and everything I'm going to tell you is due to Kamela's decision to not follow the advice of the experts, but to say, I want to study it anyway. And of course, you've gotten this advice, I'm sure, from many reviewers of your papers and grants and colleagues. Never ever think too far outside the box. So this is what ISRIP does. We have cells, we label them with radioactive amino acids, we get nice incorporation and translation. If we induce ER stress and the integrated stress response, this is reduced and AT4 is being produced. We add ISRIP to these reactions to the cells. The effect is entirely reversed. So I think ISRIP is a beautiful molecule. So I agree with Confucius here. As Lan told you, I trained initially as a chemist, and you can see beauty in many things. Twofold symmetry makes the molecule is beautiful. We have the cyclohexane. Both of these arms are identical. And because we can substitute the cyclohexane in cis and in trans, two different isoforms of the same thing. The cis has no activity whatsoever. The trans has all the activity. So we have virtually identical chemical matter as a beautiful control in these reactions. Moreover, from the primary screen for this that identified the ISRIP, it has a very high potency of 5 nanomolar. Normally, when you do these screens, you're happy if you end up in the micromolar range. So by sheer luck, serendipity, we were afforded here 3 order of magnitude advantage. And of course, that says that even though the molecule wasn't particularly soluble, and therefore the screening facility said this would never amount to anything, you don't need much in solution, right? If you have 5 nanomolar efficacy in cells, so the criteria was invalid. It has acceptable PK right out of the screen, so 8-hour half-time in a mouse. It had good blood-brain penetrance, again, right out of the screen. We didn't have to do anything. And the experiment I showed you before with Nahum Sonnenberg's lab, at that time we didn't even know that it would ever go into the brain. But we sent it to them, and they were just adventurous enough to do the experiment anyway, and it led to the development in these cognitive assays. And it showed no overt toxicity, which is now confirmed in the clinical trials. To make a long story short, we identified the target. What does ISRIP bind to in the cell? And it turns out to be this exchange factor that mediates the nucleotide exchange for the nucleotide exchange factor. So we solved the cryo-EM structure of the exchange factor in collaboration with Adam Frost. And it turns out to be this beautiful structure in a number of graduate students here. And this is the first structure we got. At the time, 2.7 angstrom was a great resolution. Anyway, it was good enough at the time. Now it could be, of course, we would not be all that happy with it. And the structure was all simultaneously or concomitantly solved by in Davis Warren's lab by Elisa. And there you sort of see the twofold symmetric symmetry, and there in the middle is our drug. The blue is the actual data. The golden halo is photoshopped for truth of advertisement. And as a twofold symmetric structure here of the molecule already sort of indicates, we have it becomes sort of intuitive what it might be doing, right? Acting as a staple of holding these two identical sides of this protein complex together. So acting like a molecular staple. And it's a very beautiful, deep binding pocket here. We did some SAR, structure-activity relationships, in the academic setting. And we basically only made it worse. So it fits so nicely already that pretty much all modifications we made didn't help. We could improve the efficacy by tenfold by putting extra halogens here. What an opportunity to make some art out of it and bringing this into the public. So this is a bronze sculpture here. I cast it from a large 3D print with a foundry over in Berkeley, my friend Sebastian Bernales. So what Calico did with it in terms of clinical development is they produced this molecule here, which they gave this awful name. So rather than having our nice ISRIP, they now call it fosigotivator, which you're all welcome to practice. And it took me weeks to learn that. And they basically made only, so I think you can still recognize the ISRIP in it. So it's still twofold symmetric. They made modifications based on our initial SAR, where they added these extra halogens here at the end to increase the affinity. The ISRIP is so insoluble because the cyclohexane in the middle allows the molecules to stack, and they aggregate and fall out of solution. So they put a little bubble in the middle, so now the stacking can no longer occur. And then they put this extra group on here, which makes a prodrug. The phosphor group is hydrolyzed off as it's being orally ingested. So it makes it orally available, increases the solubility, because once you hydrolyze that off, you're left with a little hydroxyl group here that just adds to the solubility. And this is the compound that's now in the human clinical trials. The one trial that is right now going on is in this disease called, it's a terrible disease, vanishing white matter disease, where basically it's a childhood ataxia, where the white matter, so the myelination in the brain, starts disappearing. And basically the brain starts melting. It's autosomal recessive, so the patients inherit partially defective alleles from both parents. And when they come together and these children experience some traumatic event, then basically your brain, their brain no longer all these white things here are basically lesions where the myelination no longer works. So it leads to severe neurological deterioration, progressive movement abilities, severe cognitive impairment, and 95% of the patients have mutations in the exchange factor. And they map all over the different subunits. So it's not just the active site, but it's rather making the protein complex unhappy, unstable in many, many different ways. And just under these traumatic conditions where this disease manifests itself, there's just not enough then in the brain cells to sustain the normal brain development and sustain the myelination. This is one of the patients, Enrico, who I had the pleasure of meeting. He had developed normally until he was 12 months of age. Then he had a fever that induced these conditions, rapid regression in motor function. He couldn't walk anymore. A rapid decline in cognitive ability, he wouldn't speak anymore. And genomic sequencing revealed mutations in the alpha subunit of the exchange factor. His MRI showed severe loss of myelination, and life expectancy would have been a few years. So I'm going to come back to this, to Enrico at the very end here. There's a happy ending. So what we know about this idea of ISRIP working as a staple is experimentally validated. We can take tetramer subunits of subcomplexes of the EIF2. We can add the ISRIP molecules to it and assemble them in an octamer. And in the analytical ultracentrifuge, you get a very, very clean result of the tetramers moving into an octamer peak. So this is definitely happening in the cell. There's the alpha subunit. So this is the one that's defective in Enrico here. The alpha subunit binds to the bottom of this complex. This is from the side here, and basically could also bring these tetramers together. So now we solve the structure of the exchange factor together with the substrate, EIF2. And you see that the substrate snakes over the surface of this large complex in a way that it bridges with the alpha subunit between the yellow and the blue subunit here. So in other words, complex assembly helps form the binding site for the substrate, right? So that explains why the complex assembly is useful for the catalytic activity. The catalysis happens over here, where this is a GDPase domain, and that gets sandwiched by these gray subunits here, which now pull it apart and allow the exchange reaction to happen by opening up the binding pocket. GDP can flow out, GTP can flow in, and we have accomplished the exchange reaction here. So we can explain from the structure how the exchange factor works as an exchange factor. Now phosphorylation comes in this point here. This is a serine that gets phosphorylated. This serine bridges originally in the substrate to the yellow subunit here, and when it becomes phosphorylated, this domain entirely inverts, where we now have the phosphate actually inside the folded domain, coordinated by two positive side chains here, and new hydrophobic residues exposed on the surface of this domain. And what this does is it changes the binding site as the substrate turns into the inhibitor by changing it so that now it no longer interacts with the blue and the yellow subunit, but now it goes from the yellow to the red subunit down here. So it binds on the opposite side of the exchange factor, totally changing its interaction mode. And if we extend that, this would clash with the site where the GTP exchange happens. So we could explain why phosphorylation would lead to inhibition of the reaction. So the last major message I want to leave you with, the students and the young people in the audience, is we could explain everything, right? We could explain the complex formation, we could explain the catalysis, and we can explain the inhibition. So never fall too much in love with your models, because there is a potential paradox here. And that paradox is that I've already shown you ISRP can mediate this octamer formation, complex formation, but so can the alpha subunit adding it, right? So if in the cell this complex is fully assembled, then there would be no role for ISRP based in this model to improve anything. So we need to ask the question, then, do we have a paradox? Of course, when you have a paradox, that's very exciting, right? The well-defined paradox is a perfect project. That's one of my mentors here, Ira Herskowitz, who took me under his wings when I joined UCSF many years ago. So to ask, do we have a paradox? Enter Michael Schoff, a new graduate student, here with a cat. And Michael built this fantastic reporter in which he allowed us to ask this question directly: how much of the complex is assembled in a living cell? And he built this with putting two fluorescent proteins in proximity, that when the complex is assembled, you can get fluorescent energy transfer between the two dyes, and you can read that out fluorescently. And he put a regulated degron on the alpha subunit. So we could take the alpha subunit, add a little chemical, the induce degradation here, so we can take the alpha subunit away experimentally as we please. Now this reporter works fantastically, much better than I ever had expected. So if we now titrate into cells that has the reporter for the response, and if we titrate in the degron and you degrade the alpha subunit, you see that now the complex falls apart and the response is induced, right? We've inactivated ERF2, the exchange factor here, as expected. If we now add ISRP to these cells, we put the thing back together. Even in the absence of the alpha subunit, forming the octamer is sufficient. It has all the components we need for substrate binding and GTP exchange. So that's consistent. Now, if you look at the fluorescent reporter and look at the actual assembly, you see that as we titrate in the destruction, or we cause the destruction of the alpha subunit, now the complex falls apart, the FRET signal goes down, and as we add ISRP, the octamer comes back together, even in the absence of the alpha subunit, and we restore the complex. But the important thing here is this black line up, the control up here, where we did not degrade the alpha subunit. And you see it's the same plateau. So in other words, the complex is fully assembled in the cell. So now we need to explain how, under these conditions, the inhibition happens. So how can ISRP counteract the inhibition of the exchange factor by the phosphorylation event? And this is the last big data slide here. It's a very simple experiment that resolves the paradox. We are just doing a pull down of an in vitro reaction here. You see, if we pull down on the exchange factor, we bring down all three subunits of the substrate. If we add the phosphorylated form to this reaction, you see it binds, but it kicks off the substrate. So the two are not simultaneously bound. And if we add ISRIP to this reaction, you see that now the inhibitor no longer binds, the phosphorylated form, but now the substrate can bind again. Since these two bind at opposite sides of the complex, we must have allosteric communication through the complex, where ISRIP and substrate go hand in hand. Right? And we can show that with adding the inhibitor, we actually kick out the substrate. Now, if we had this communication happening, then we should have seen it in the cryo-EM. And the fact is, we did, but we didn't pay attention to it because it's a quite subtle change. So the current model, not exclusive with the fact that we could also have mediation in some cell types where ISRIP will simply function as this molecular glue, is that ISRIP changes the conformation of the protein, biases it to an active state, under which EIF2 the substrate can bind and be subjected to its GTP exchange, whereas the phosphorylated form, by binding on the opposite side, would cause an inactive state and therefore now compete or allosterically affect binding of the substrate and binding of the ISRIP. And we can in fact see that in the structure. So here's the active state. If we bind the inhibitor to it, that's what it looks like. And if we superimpose the two, we now end up with our Mariposa butterfly model here, where you see the subtle hinge movement between the two domains that communicates the ISRIP and EF2 binding phases up here with the bottom phase. And we can go one step further. We can, Rosalie Lawrence, a recent postdoctoral fellow who's now has her own lab at UCLA, identified discrete residues in this one switch helix in the center here that go back and forth and provide very nice molecular definition in this conformational change. This has been further expanded now by Advait and Udit at Altos, where we now have an entire communication chain of residues at the interface that flip back and forth between the two different states. And we can make mutations now in these various things and lock it into one state or the other, and everything pretty much works as expected, which gives us a lot more pockets and surfaces here where we can potentially target with small molecules and affect the equilibrium of this exchange factor. The changes are incredibly subtle. So in the ISRIP binding site, we have a few angstrom that make all the difference between the active and the inactive state. And this basically is at the crunch of the action of this drug. So important take-home lessons, what I would like to leave you with, is that inhibiting the cell stress pathways is a clinically important approach spanning a wide range of different diseases. Many more than just the cancer example and the cognitive defects I showed you. That we identified ISRIP as a potent first drug-like molecule to block the integrated stress response. To understand ISRIP's molecular mode of action in atomic detail. We showed ISRIP's efficacy as a cognitive enhancer and identified the integrated stress response with that as a druggable pathway and its inhibition as a promising avenue to combat cognitive disorders. And we have this amazing efficacy in a broad spectrum of cognitive loss and brain health. And the important thing here is that we had no idea where we were going. right? It's a meandering path of discovery. And this all emerged as a result of curiosity-driven, disease-agnostic basic research. And that's what we should hammer in at every occasion we have to our funding agencies and to our institute leaderships and so on and so forth. That leaves me with showing you a picture of Israel here. So Enrico now has been for 3 months in the clinical trial with this calico drug. And I told you he couldn't walk anymore. He couldn't. And we just met him. We had lunch with him. He has an amazing vocabulary, and he's running around like a normal child. The clinical trial isn't read out yet. So this is information from the parents, private information. So we are eagerly awaiting that, going from an N of 1 to an N that's a little bit larger and more believable than this one story. But it's heartwarming. So thank you so much for your attention, and happy to take some questions.
So, Peter, so that was fantastic, and sort of teach us how to think about basic cell biology to diseases. Stress response, including integrated stress response, obviously has very important physiological functions. So when we think about targeting diseases, would there be any long-term liability impacting how body normally deal with stress condition or just physiological condition? Meaning that would there be long-term possible side effect?
Yeah.
A related question is, if we think about disease, obviously this particular disease had mutation in the subunit. But if broadly you're saying disease agnostic type of thinking, how would you pick other diseases to target? For example, we study Alzheimer's disease.
Yeah. I wouldn't pick them, right? We are the data leading us to these applications. For the IA1 story, the unfolded protein response story in cancer, it's basically, so Sun Wen sitting here is part of our collaboration with Genentech Group. We are basically studying the basic cell biology and then applying the knowledge that we are getting and seeing where can we be efficacious in a disease setting. In this case, we do it with the experts, right? We do it with the cancer group at Genentech, who does this for a living and who knows what cancers are best to look at in the clinical trials and how to basically block best the IA1. Is it this inhibitor or is it other modes of getting rid of this activity? Long-term effects, we need to see what happens. I told you that the heterozygous mutant has these cognitive improvements, but that the homozygous mutant is the mouse is dead. So we need the integrated stress response. But the way this mechanism worked out is that if we have the integrated stress response turned on very strongly, so we make a lot of phosphorylated form of the EIF2, then it kicks out the SREP, right? Because of the allosteric mutual binding activity. So it's limiting itself. This is fortuitous. We didn't design this in. This is what we found from the results. So we just need to put this, walk the walk and go through the different steps of characterizing the system properly. We've done ISREP studies in mice through a lifespan of mouse, and we see no defects. We see no added vulnerability to viruses, for example, which one of the kinases is sensing. That doesn't mean that this couldn't occur in a human setting, right? And this, I think it would be for Enrico here, it gives him a chance on life, but he probably would need. We cannot take the mutants away with this approach, so he probably would need to be on this drug permanently. for traumatic brain injury, where we consider perhaps a one-time event, right, that causes the problem, and we can use the drug as a reset button. We have a much different way in the clinic. But the clinical trials haven't been done because the industry works differently from the way our brains work.
Okay. Thank you for the wonderful talk. I mean, it's super cool to see this child can walk again. Just for curiosity, what about this kid, other learning skills, or even he become much more smarter than before?
Yeah, that's right. I was amazed by his vocabulary. For now, how old is he? 2? Almost 3. So we played ball, right? He can catch things. He can communicate very, and given the fact that he was very, very sick before, it's truly remarkable.
Hi, Peter. Very nice talk. So just curious, when you give the drug to the people, so is a lifelong treatment, or can be stopped in the middle or once they get recovery? Based on animal experiment, it looks like if you treat the mice for a certain period, you stop for a certain period, but still the effect can last very long. I just curious at that time, because at that time the stress response is still there without the drug, right? But still the mice can still have the better memory or learning abilities.
But we don't know. In the traumatic brain injury, we don't know when the response is, the stress response is still there, right? The same way in the traumatic brain injury, you have a one-time insult, and just resetting it may be sufficient. In other cases, I mean, Down syndrome, we could not take this method would not take the extra chromosome away, right? So the stress response would stay on, right? So there you would need a different treatment regime.
Peter, as you were saying, the lack of toxicity of ISRIB is really amazing. But I was wondering if approaches have been tried to inhibit some of the upstream kinases leads to EIF inhibition.
Yeah, has been tried. So if you inhibit PERK, it's severely toxic, because now you take the response. In that case, you get pancreatic toxicity. So that was stopped in the clinical trials. So GSK had developed some inhibitors there, because the integrated stress response in its strong mode, where ISRIB no longer works because it's got kicked out, you have too much inhibitor buildup, is important in the endocrine pancreas for glucose insulin homeostasis.
Thank you.
Yeah, Peter, thanks for the talk. So I just curious, is there any side effect for the kid currently for the 3 months treatment? I mean, side effect.
But we have not seen any.
No.
We don't know of any. No overt toxicity has been reported.
I think everyone's already trying to buy their own, and if there's no
Can buy it from Alibaba, yeah.
Thank you, Professor, for the talk. And I have one question related to the treatment to neurodegenerative disease. So, according to my understanding, the medicine can refresh the core neurons, but there's lots of neurodegenerative disease that has a lot of death neurons. So does that mean that if we can maintain the fresh and young activity of the current neurons, it is enough for the cognitive things, and we don't need to care about those death ones? Or there's a kind of threshold that if it's that so much, the medicine cannot help?
Yeah, I don't think we know, right? And it may have been very fortunate that Enrico was diagnosed so early in life, right? Because the young brain is much more flexible in able to regenerate than an older brain. And that is, it's a rare disease, but we don't know really why it's rare. It could be, the allele could be in many places where it hasn't been diagnosed, right? He was just lucky that he and his parents got sequenced so fast at Stanford. They had a pediatric neurologist who immediately diagnosed it or thought that sequencing was appropriate to then diagnose it with molecular definition. And most of the world wouldn't have access to that, right? So we don't know. We'll find out. And in Alzheimer's, the same thing, right? I mean, once the cell is dead, we are not going to bring it back to life. So if it's a pre-treatment, like we do routinely now for statins, right, for cholesterol synthesis, may be preventive for things like that from developing more severely. So early treatment. We're missing the biomarkers. We're missing the we have a lot of work to do for everybody. It's a wonderful. It's job security.
Any more questions?
Thank you. So I was very much amazed by the 3-year screening, but may I ask a naive question? How do you came up all those candidates, like without nowadays the AI tools, for example?
By the screening, and it wasn't all the candidates. We had a library of 100,000 compounds of random chemical. There was no AI. There was not even natural intelligence. It was just brute forcing through a random collection of small molecules to see what chemical matter would give us this physiological effect in the cells. And the reason why it took 3 years is because we've never done it before, right? And you run into all sorts of issues of when you go from a small scale into a high throughput setting, it requires a lot more work than if we had known how difficult it was, we may not have done. Camilla might not have agreed to do the experiment. So it was good that we didn't know. Yeah. So we ran planning on 3 years. I thought it's a Sunday afternoon.
Any more questions? Okay, maybe I ask one last one. For Enrico's case, do we know if ISRIB actually enhances complex assembly or biasing towards the A state?
No, the mutations were one of the mutations was known, the other one was totally novel and no in vitro. That hasn't been reconstituted in a model.
All right. If no questions, let's thank Peter again. We'll have a reception on the 5th floor.